We've been talking about carbon molecules, and we went over structures for carbon and its ability to form large polymers. We talked about carbohydrates and we talked about lipids. So the next thing we need to talk about is proteins. Now, protein molecules have very diverse functions within the body. They can be digestive enzymes or any kind of enzymes. They're involved in transport, they're involved in structure, hormones, defense mechanisms. Your muscles contract and organisms move through the use of myosin and actin, which are protein molecules, and there are storage molecules. The egg white, albumin, is a storage molecule that allows the embryo of the chicken to actually grow. That's why they are so high in proteins. Now, proteins are made up of monomers of amino acids. And amino acids remember what this group is right here? That's the amino group. Oh, sorry. This is the amino group. And then you have a carbon, alpha carbon in the middle. And then you have another carbon that has a carboxyl group on it. So it is an amino acid. And those form into polymers of amino acids called proteins through a peptide bond. Remember dehydration? So that peptide bond here gives off water, and that forms the long polymers of protein molecules out of amino acids. Now there are 20 some odd amino acids that are important to man. Now I don't expect you to memorize each one of these amino acids, but what is important is what is the R group. This gives the amino acid its different chemical characteristics. So whether it's a non-polar, a positive charge, polar molecule, negatively charged, those are all determined by the R group. Now that R group, remember, is on this amino alpha carbon on the amino acid. Now, proteins are very complex molecules, and they have different levels of structure that are important. The first order of structure is the actual sequence of amino acids. That is the primary structure, and that primary structure, we'll find out, is actually determined by the genetic code. The genetic code tells us what amino acid is going to be put into different place in that polypeptide chain. The next level of structure is secondary structure. And there are basically two structures: alpha helix, which are coiled, or beta pleated sheet, which are areas of a protein that have long, flat sheets. And those are kept in that structure by these hydrogen bonds. Now remember, we talked about hydrogen bonds being important. Here's another place where hydrogen bonds actually help determine the secondary structure of a protein. You have the tertiary structure, is how it's folded back upon itself. And this is going to include not only hydrogen bonding, but also things like hydrophobic areas where there's amino acids that want to shed water, hydrophilic areas, disulfide bonds, bridges between, and ionic bonds forming between the R groups of the amino acid. And then the quaternary structure is what you fold a protein molecule several together to make a protein. Now, that quaternary structure is going to be very important. The structure of hemoglobin is determined by the interaction of the 4 peptides that form up hemoglobin, 2 of the alpha hemoglobin and 2 beta hemoglobin, around an iron molecule. And if you change one amino acid in that one of those peptides that form the hemoglobin molecule, it will no longer function as normally in hemoglobin. And you get a disease, what is known as sickle cell anemia. The last major organic molecule we're going to talk about is nucleic acids. Now, nucleic acids are the information molecules of the cell. These are the components of nucleic acids are what make up the DNA, the gene, and the information that gets taken from the gene and made into the different parts of the cell for use in the cell through the use of RNA, ribonucleic acid. And you have different bases in it. So the DNA makes up the chromosome. That's the information hub for all of the cell. That's where its information on how to make everything it needs for that cell to survive is contained in that DNA. The structure of a nucleic acid is that it has a nucleoside base, either purine or pyrimidine base. It has a phosphate group, and it has a sugar group. Now this is ribose. This would be RNA. In deoxyribose, this here is missing, has a hydrogen there, and it becomes deoxyribose. That is one of the 2 differences between DNA and RNA, is the structure of the sugar molecule that's used in the sugar backbone. The other difference is in the nucleoside base. Both DNA and RNA use adenine, guanine, and cytosine. DNA uses thymine, RNA uses uracil. And these bases are complementary. That means they bind to each other, and they won't bind to anything else. Adenine always binds to thymine, or in the case of RNA, it would bind to uracil. Guanine always binds with cytosine, and that is a hydrogen bond that goes between those different structures. And that base is only going to be complementary to the base that it's only going to hydrogen bond to the base that is complementary to. The structure of DNA, you have the nucleoside bases, you have the sugars involved, and you have a phosphate backbone. And notice that you have the hydrogen bonding here between the nucleotide bases. Comparison of RNA to DNA, these are the 2 main information molecules in the cell. DNA is the genetic code that's in your genes, is in your chromosomes in the center nucleus of the cells. It is deoxyribose as a sugar, whereas RNA has ribose. Bases, the only difference is the substitution in RNA of uracil for thymine. And the structure of DNA is a double helix. That means that you have 2 strands of DNA. They bind to each other by complementary hydrogen bonding between the complementary bases, and they actually have direction. Information runs a specific way based upon that DNA binding, that DNA molecule. And we'll get to that much later when we talk about DNA replication and also how you get information out of the genetic code to make proteins. RNA is single-stranded. And very seldom do you find RNA which is bound back on itself. There are some RNA structures that are that way. Most of the cases of RNA, and most of the RNA is single-stranded. Okay, so that ends the lecture. If you have any questions, make sure you post them up. Make sure you read your chapter. Make sure you do the discussion. Make sure you do your homework, and take the test at the end of when you're ready. But make sure you study before you take the test. It's always best to do that. Okay, have a good one. Bye-bye.